We're going to talk about beta oxidation of fatty acids. So beta oxidation of fatty acid is the catabolic process by which fatty acid molecules are broken down and generates acetyl-CoA. So let's talk about the type of reaction. It is a catabolic reaction, that means breaking down type of reaction. And the site of reaction is mitochondria, and to some extent it also takes place in the peroxisome. However, there are some differences in mitochondrial versus peroxisomal beta oxidation. So let's just look at a mitochondria and try to understand the process of beta oxidation in bit more details. So here is a mitochondria, and you can see a zoomed in view. Now inside the mitochondria, fatty acyl-CoA, in this particular case the example is palmitoyl-CoA, a 16 carbon fatty acid. The fatty acyl-CoA ultimately gets converted to acyl-CoA, in this case myristoyl-CoA, and it releases acetyl-CoA. The acetyl-CoA that is released can be channeled into TCA cycle, and ultimately the high energy compounds that are produced from the TCA cycle such as NADH and FADH can be utilized in the electron transport chain to generate ATP. That's the goal. But looking at the beta oxidation of fatty acid, we have to consider few aspects, and these few aspects are: where does beta oxidation takes place, and we have a clue about that. Second is like under what condition beta oxidation takes place. Third is what are the enzymes that helps in beta oxidation process. Obviously inside the body any reaction or any biochemical pathway would be enzyme driven. So we have to understand what are the enzymes that are regulating this pathway. And lastly, how beta oxidation process is regulated depending upon the demand. So let's begin. Now the metabolic state is the key aspect or the key circumstances under which fatty acid oxidation is favored. For example, in the fed state, when the body has too much of energy, then fatty acid anabolism is preferred. At that state, body would prefer to store the fat in adipose tissue. But in fasted state, the fat would be broken down and it would be utilized to generate energy, right? So let's look at this process in bit more details. So let's assume we are right now in a fasted state, and in the fasted state, inside the adipose tissue, the triglycerides would be broken down to generate free fatty acids. Now these free fatty acids would be carried into the liver, and in the liver hepatocytes, the mitochondria of liver hepatocytes, they would be broken down and they would be oxidized, and the ultimate goal is to generate energy under the circumstances of fasting, because in the fasting we don't have supply of food, so glucose level is limiting. Now fatty acid would be ultimately converted to acetyl-CoA inside the mitochondria, and the enzymes which are important are present in the mitochondrial matrix. So let's look at the mitochondrial matrix to understand this process in a lot more details. But there is a basic question: how does fatty acid gets inside the mitochondria? Because the mitochondrial inner membrane is impermeable to the fatty acid. So it turns out there are transporter mechanisms present in the mitochondrial inner and outer membrane, which helps the fatty acid to get inside. So there is a small molecule known as carnitine. The carnitine makes a conjugation with these fatty acid. For example, in this case it's a palmitoyl carnitine, and this adduct is actually transported by carnitine palmitoyl transferase 1, or commonly known as CPT1, present in the outer mitochondrial membrane. And ultimately, using a carnitine acyl translocase channel, the palmitoyl carnitine gets inside the mitochondrial matrix. Now with the help of carnitine palmitoyl transferase 2, the palmitoyl-CoA is released and the carnitine molecule is recycled back to the cytosol. Now the palmitoyl-CoA, which is inside the mitochondrial matrix, is the starting material for beta oxidation process. Ultimately, in an enzymatic process, This palmitoyl-CoA would generate acetyl-CoA, several molecules of that. We would look at the count of how many acetyl-CoA would be produced. So stay tuned till the end of this video. Now inside the mitochondrial matrix, this is our starting material, palmitoyl-CoA. Now notice that the carbon atom, which is just near the functional group, is the alpha carbon, and the carbon nearby is the beta carbon. This process is known as beta oxidation. So a lot of changes are going to happen in the beta carbon. So we need to take a look at the beta carbon at each step. Palmitoyl-CoA would be converted to trans delta 2 enoyl-CoA. As you can understand that there would be a double bond formation between alpha and beta carbon. In short, we can say hydrogen number is reduced. In other ways, we can say there is oxidation taking place. And the enzyme that triggered this process is known as acyl-CoA dehydrogenase. As you know, these dehydrogenase enzymes can perform oxidation reaction, right? So from trans to enoyl-CoA, it would be converted to beta-hydroxy acyl-CoA. And again, if you look at the beta carbon, a hydroxy group is attached right now. So it is oxidized further. And the enzyme is enoyl-CoA hydratase. Point to be noted that at the first step, FADH2 is produced. Later on, I'll tell you what these FADH2 and NADH is used for. Then, beta-hydroxy acyl-CoA gets converted to beta-keto-acyl-CoA, where the beta carbon is now having a carbonyl group, which is even more oxidized version than an alcohol, right? And the enzyme which is triggering this process is known as beta-hydroxy acyl-CoA dehydrogenase. Again, another dehydrogenase enzyme. So majorly there are two oxidation steps in this whole beta oxidation process, and every time the beta carbon is changing. In this process, something is reduced, which is NAD. NAD is reduced into NADH. NADH and FADH2 would be utilized later on in the electron transport chain to generate energy. Ultimately, a acyl-CoA molecule would be produced along with the acetyl-CoA. So, 2 number of carbon is reduced from this long chain. So, if this chain was 16 carbon long, now it has become 14 carbon long because 2 carbon acetyl-CoA is moved out. Now, the acyl-CoA, in this case the myristoyl-CoA, can be acted further up by other enzymes. I forgot to mention that the last step is catalyzed by thiolase. Now, these 4 enzymes are the key enzymes for beta oxidation. Now, look at the beta carbon, how at different stages there are changes in the groups associated with the beta carbon, right? So that is why it is the beta oxidation of fatty acid. So let's assume this is the 16 carbon palmitoyl CoA or palmitic acid. It would be eventually broken down into 14 carbon, then 12 carbon, and gradually it would release all the molecules, and the cycle would round about 7 cycles. It would eventually give the product would be 8 molecules of acetyl-CoA, 7 molecules of FADH2 and 7 molecules of NADH. All these NADH and FADH2 can be further utilized in the electron transport chain. Now, strictly speaking, all these 4 enzymes that are important for fatty acid oxidation, all these enzymes are not individual entities. They actually form a trifunctional protein complex, which is a heterooctamer having 4 alpha, 4 beta subunits. Now, the beta subunits contain the activity for thiolase, and the alpha subunit has a dual activity for the hydratase and the hydroxyacyl-CoA dehydrogenase. So, dehydrogenase and hydratase activities are intrinsic properties of the alpha subunit of these big trifunctional proteins. Any fatty acid which is greater than 12 carbon are processed by these trifunctional protein, and this kind of depiction of 4 individual enzymes are a bit primitive. Now let's try to understand what happens to this acetyl-CoA and just an overview. So this acetyl-CoA would ultimately be broken down into acetyl-CoA. Each time the acetyl-CoA is produced, it can be channeled into the Krebs cycle. Ultimately, in this process of converting the acetyl-CoA to acetyl-CoA, several molecules of NADH and FADH2 would be generated, and also the acetyl-CoA which has entered into the Krebs cycle would also produce NADH and FADH. All of these NADH and FADH2 would be channeled to the electron transport chain. They would donate their electron to the electron transport chain, and proton would be pumped into the intermembrane space. As a result, using the proton gradient, the ATP synthase would generate ATP. Now this is the key goal of beta oxidation. Beta oxidation's key goal is to break down the fatty acid in the time of starvation, when the body needs fatty acid breakdown to generate energy. Now we would learn about a little bit about peroxisomal beta oxidation and try to appreciate how it is different from a mitochondrial beta oxidation. Now all the enzymes that carry out the beta oxidation process in peroxisome is also very similar, but only difference is, in case of peroxisomal beta oxidation, the FADH2 is not channeled into the electron transport chain. Rather, it is utilized to reduce oxygen into hydrogen peroxide, and that's how peroxisomes produce hydrogen peroxides. And for peroxisome to produce hydrogen peroxide, beta oxidation is super important. So now we can appreciate the same process in two different organelles and how they are different in a certain aspect. Now let's try to understand the regulation of fatty acid catabolism process. So fatty acid catabolism process is under regulation of hormones, transcriptional regulations and regulations at several different levels. We would restrict our discussion to hormonal regulations and transcriptional regulations. So we looked at the first and the foremost step is to transport fatty acid from the cytoplasm to the mitochondrial matrix, where the fatty acid would be broken down. If the fatty acid which is present in the cytoplasm is not transported to the mitochondrial matrix, it won't be broken down and utilized. So this transport process is regulated. By whom? By several hormones such as insulin and glucagon. It turns out malonyl-CoA is an inhibitor of CPT1. Now how malonyl-CoA is produced? Malonyl-CoA is in turn produced by acetyl-CoA, and from malonyl-CoA fatty acid biosynthesis can take place. Now malonyl-CoA would be only generated when there is sufficient amount of acetyl-CoA or in the fed state. Now acetyl-CoA carboxylase triggers this process. Acetyl-CoA carboxylase is active when insulin is there, and we all know that insulin is there when there is a fed state or when there is enough amount of glucose. So presence of insulin ensures that acetyl-CoA carboxylase is active, acetyl-CoA is converted to malonyl-CoA, and malonyl-CoA shuts off the transfer process of fatty acid from cytoplasm to the mitochondrial matrix. Rather, malonyl-CoA inhibits the CPT1 and it triggers the fatty acid biosynthesis pathway rather than the fatty acid breakdown process. So in the fed state, we would understand that fatty acid biosynthesis is favored rather than the fatty acid breakdown. In contrast, in the fasted state, fatty acid breakdown would be favored rather than fatty acid biosynthesis. So we understand how hormonal level can regulate the